So let's bring out some other tools. So I have a diffraction grating. So this is a piece of plastic with, this says 500 lines per millimeter on it. So these lines are too small to see, but what we can do is use lasers to interrogate what's going on. So here's an object in real world that's too small to see, and we're going to use physics principles, just like what we saw, to basically tell us what's going on, to see what is normally invisible. Okay? So if this is 500 lines per millimeter, what do we expect to see when we shine a laser through this? What is it? Light. We're going to see light. So here's our laser. Can we see the green spot in our little cove up there? So now I'm going to put my diffraction grating in front of it. What do we see?
More green spots.
More green spots. Okay? That's called diffraction. If you shine a laser through a diffraction grating, you get diffraction. And the location of those spots tells us about the spacing of the lines. Okay? So 500 lines per millimeter. And now I'm swapping that out for another diffraction grating that says it has 1,000 lines per millimeter. What do we expect to see if this diffraction is in bizarro world?
Either half or twice.
It's either half or twice. Let's take a little ballot. Who thinks it's going to be wider space spots in our experiment? Who thinks it's going to be narrower space spots in our experiment? Okay, so if we have 500 lines per millimeter versus 1,000 lines per millimeter, which is the smaller object? Right? 1,000 lines per millimeter is a smaller object in real space. What happens if we go into bizarro space or reciprocal space? Larger spacing. Okay, there's 1,000 lines per millimeter. Here's 500, and here's 1,000. 500 and 1,000. So we get a reciprocal relationship. The smaller the object in real space, the wider the spacing in reciprocal space or bizarro world. Okay? What happens if I take two of these diffraction gratings and turn them 90? So now I have one set of lines that's going this way, another set of lines that's going this way. What's going to happen when I shine my laser through? We're going to now get an array of spots. Right? So we have diffraction in the up and down direction. We have diffraction in the left to right direction. And I have a couple different diffraction gratings with different spacings. Okay, so we can interrogate shapes in 2 and 3 dimensions using tools like diffraction. Okay, what happens if I have a couple of diffraction gratings that are offset from one another? So I take we have a 2-dimensional diffraction grating, and then I put another 2-dimensional diffraction grating, and if my laser holds up, I get lots of spots, and if I rotate one relative to the other, I get all kinds of star patterns. Sorry for those of you that are on the wings, but I can't figure out anywhere else to do this. Is that better? Okay, that's 2 aligned diffraction gratings, and then if we offset them, we get all kinds of dancing stars. Okay, so we can use, question, yeah? That is a fantastic question. The question is, what happens if you change the color of the laser? As it turns out, I collect lasers. Here's a green laser. So let's see if I can hold two lasers and a diffraction grating in one hand. Here's our green laser. Here's a purple laser. And do I have the dexterity to pull this off? Okay, the location of the spots depends on the wavelength of the light. I also have a red laser, but I don't have the hands to pull that off. Okay, so there's a relationship called Bragg's law that tells us about there's a very clear mathematical relationship between the wavelength of the radiation and the size of the spots in real space, the size of the lines in real space, and the location of the spots in diffraction space. Okay. So now let's take a look at what we've done so far. We did our light diffraction experiment. We shone light at an object like Grace. We captured that scattered light with our lens, and then we made an image by collecting that scattered information. So now if we want to probe objects that are at smaller size than the wavelength of light, we're going to use that same principle, but some of the pieces of hardware change. Instead of light, we're going to use X-rays. So there are ways of making X-rays. You can basically use a filament in a light bulb and basically crash it into a piece of metal, and you get X-rays. So now we're going to shine X-rays at an object. So now we're not going to shine X-rays at an object that's the size of a human. We're going to shine X-rays at an object that's the size of a protein. Okay? So we're 10 to the 10 orders of magnitude smaller than a human. And what we get is an X-ray diffraction photograph. Okay? We just saw laser diffraction with 1,000 lines per millimeter. We need to go 5 orders of magnitude smaller than that. So we're not going to use visible light. We're going to use X-rays, but the principles are the same. We're going to look at those spots the same way we saw those spots as generated by the lasers. Okay? So now it turns out X-rays don't bend very nicely question from my spouse.
It's a softball. What's the difference between light and X-rays, and why would you use X-rays instead of?
Right. So when we went, so the question is, why are we using X-rays and not light? So it turns out the object you're interrogating, the information you want from the object you're interrogating, is related, the information you can get out of it depends on the wavelength of the light you shine at it. So if you're shining visible light at an object, the most you can see is like 400 to 700 nanometers, sort of on the order of the wavelength of visible light, which is 400 to 700 nanometers. That's not good enough if you want to see atomic distances, if you want to see proteins, if you want to see stuff that is still 3 or 4 orders of magnitude smaller than that. So we're going to use electromagnetic radiation, but it's not going to be visible light, it's going to be X-rays. So X-rays, again, I mentioned a carbon-carbon bond is about 0.15 nanometers in distance. The X-rays we use in our lab are about that same size, within a factor of 2 or something like that. So we're, again, using the wavelength of radiation that interrogates the information we're trying to get. And in this case, we want to know where atoms are, and visible light doesn't have enough high frequency to do that. Okay? So now we're going to use some mathematics. And this mathematics is, physicists have worked out these principles for hundreds of years. And the mathematics are complicated. They're called Fourier transforms, but they're straightforward. It's the Fourier transform, or the lens, is how we get from diffraction space, or bizarro world, back into real world. And so those mathematics we understand due to the hard work of physicists over the centuries. And when we do that, we take our diffraction pattern, we do our transformation between bizarro world back to real world, we get an image that comes out the other side, and here's an image of a protein. So again, we're at 10 to the 9 orders of magnitude there in amplification, but we can actually also look at atomic distances, carbon-carbon bonds. If we want to do chemistry, we can shine X-rays at proteins, and that tells us where the atoms are. And if we know where the atoms are, we know how to do interesting chemistry. Okay? So we're using the tools of physics to answer questions in biology and solve them in chemistry. So those of you in school often treat biology as different from physics, which is different from chemistry. But when you get to real world, it turns out all of that stuff mixes together in this bizarre world that we visit with diffraction. Okay, and we are, of course, standing on the shoulders of giants. This is an X-ray diffraction of a protein crystal. This is an X-ray diffraction image of DNA. Oh, and I got chopped off a little bit at the bottom. But Rosalind Franklin took this X-ray photograph, and she was the most gifted experimentalist in her field in her day. And this is basically an experiment that brought lots of people from the world of physics over into the world of biology. Okay? And in Bizarro World, right, so what do we know about the structure of DNA? We know Rosalind Franklin pulled out a fiber of DNA, shot X-rays at it, and got this pattern. What do we know about the structure of DNA? Does that look like the structure of DNA? So this is the Bizarro World representation of DNA. So in the Bizarro World representation of DNA, a long distance here is a short distance in our world. So DNA has bases. The bases stack together on top of one another, and that spacing is 0.34 nanometers. Every base in the 3 billion base pairs of your DNA stacks on top of one another. That spacing is repeated over and over and over again, and that small spacing, the stacking of DNA bases, is represented by this big smeary stuff here. That's in reciprocal space. That's 1 over 0.34 nanometers. We can measure that distance. And again, Bragg's law says we can get from reciprocal space or Bizarro world back into the real world. Okay? So big is small and small is big. So this long spacing here tells us about close spacing in the structure of DNA. Okay? In Bizarro world, a cross is a helix in the real world. So Francis Crick looked at this image, saw the X to somebody that studied helical diffraction like Francis Crick did. The X immediately means there's a helix in there, and that was one of the insights that allowed Watson and Crick to make a model for the structure of DNA based upon these and other evidence. Okay? And it turns out I don't have this demo going. I didn't get it. I got too distracted when I was getting going. But I will just explain it. So when we go from diffraction space and go back into real space, it turns out those X-rays that we shot at our protein, they interact with the electrons. So the result of an X-ray diffraction experiment is the location of the electrons in the protein.
Excuse me, what is the shape of the helix?
What is the shape of a helix? We're just about there. We're almost to the point where we're done with the physics and we're headed into the biology. But we're getting there, we're getting there, we're getting there. Okay, so now we need to know some biology and some chemistry before we can answer the questions we're trying to answer, like can we cure the common cold? So proteins are made of amino acids. And amino acids have their own personalities. Some of them are very social. They want to meet their opposite type. So we have negatively charged amino acids who like to interact with positively charged amino acids the same way high school boys and girls like to interact. We have grumpy amino acids that basically want to be left alone. Okay? They do not want to hang out with other amino acids, and they more or less want to be left alone. And then we have amino acids that like to form gangs. Okay? So each of the 20 amino acids has their own personality, And when you make a polypeptide, you string together these 20 amino acids in different combinations in a linear way, and the personalities of the amino acids determine the folded shape of the protein. Okay? We need to know one more thing about protein. Protein's made as a linear string, and then that linear string does stuff based upon the personalities of the characters that are encoded in that particular amino acid, in that particular polypeptide chain. So there are some things called secondary structure where we have these helices. So sometimes the polypeptide likes to spin up into a helix. Sometimes it likes to form these sheets, these corrugated sheets. And then those secondary structure elements come together to make tertiary structure. So there's biology and chemistry that drives it, but basically the personalities of these different amino acids determine how that linear polypeptide folds up. Okay? So now is time for you guys to do your first model building exercise and grab your polypeptide chain, and if we put it on our left index finger and wrap towards us and go towards the end of our index finger, that should make a right-handed helix that we're going to call an alpha helix. Anybody successfully make their first alpha helix? There's an alpha helix over there. It should be right-handed, which means as you spin around, you should curl around. It should follow your right hand as you trace it around. Okay, if you made a left-handed alpha helix, that's okay, but they're rare in nature, and we're trying to be extremely accurate with our pipe cleaners here. Okay, the next secondary structure element we want to make is a beta strand, and I don't know how I'm going to show this. So a beta strand is like a corrugated piece of cardboard. Every other amino acid, up, down, up, down, up, down, up, down. So the backbone goes up and down, and it's called a beta strand or a beta pleated sheet, and it kind of looks like corrugated cardboard. Okay? See if you can make a beta strand by making some corrugations in your polypeptide chain. And so now you've made the primary building blocks of a polypeptide, of a protein structure. So we have our primary chain, that's the linear sequence. It folds up into these secondary structure elements, and then the secondary structure elements pack against one another based upon the personalities of the different amino acids, and that determines the fold. So you start with any arbitrary sequence of amino acids, and then you fold up into a specific shape, and that's determined entirely by the personalities of the amino acids that are encoded in that polypeptide chain. How are we doing? Anybody make, this is a very famous polypeptide fold. Anybody who made that should come and sign up in my lab immediately, please. Here's an effort I made. This is it's an approximation of this. This is called a beta alpha bait barrel. It's a beta strand and then an alpha helix, and then another beta strand and then an alpha helix, and then another beta strand and then an alpha helix. So you have 8 beta strands in the middle and 8 alpha helices around the outside. I think I needed quite a few pipe cleaners to make that. If you need more pipe cleaners, we have more. Okay. So one of the things I want you to notice is when you get a folded polypeptide, different places in that linear sequence end up in the same physical space, right? So it turns out in a fold like this, this loop and 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 this loop, which are widely spaced in the primary sequence, end up in close physical proximity. And the personality of the amino acids that are in these loops, they want to do chemistry. They want to break bonds. They want to bind stuff and cut stuff into pieces. Okay? So that's how life works at the chemical levels. Your DNA encodes polypeptides. Polypeptides fold up and do chemistry that the cell needs to get stuff done. Question?
What's the difference between a polypeptide and protein?
Yeah, a polypeptide is a string of amino acids that then folds up into a protein. So they're the same chemical species, but in my field, you normally think of a